Hello and welcome back to part 2 of human growth and development. So in the first part we had gone through the process of fertilization, and then we talked about how the conceptus is going to make its way through the uterine tubes into the uterus and implantation, and then begin the embryological period. And so from fertilization to implantation, that is the pre-embryonic period, and then now we have implantation, which is roughly about the second week, to the end of the 8th week is the embryological period. And so I just kind of wanted to talk about something. So stem cells, you hear a lot about them. We know that stem cells, these are undifferentiated cells, that they are programmed to divide through mitosis. And so when we looked at this period of cleavage, which is part of the pre-embryonic period, notice how all the cells are the same. So these stem cells are what are considered totipotent stem cells. So think totally potent stem cells. So we know that you can take this and split it and you can create two human beings from that. And so what we mean by totally potent is that those cells can become any cell in the human body. And also something we're going to talk about this lecture is the extra-embryonic membrane. So things like the placenta and the amnion and the umbilical cord. And so down here we got some differentiation. And so what differentiation is, meaning they visually look different. Some genes are going to be turned off forever, some are going to be turned on, and we're going to have a difference in function. And so you can already visually see that once we got to that blastocyst stage. Remember that blastocyst stage is the period of development where we get implantation. And so I realize it's just a model, but it's showing you that, okay, these are all yellow cells. And then look, now we have dark orange cells. So that we're already getting some differentiation even before we get implantation. And so these cells that are part of that inner cell mass, they're called pluripotent stem cells, meaning they have the potential to differentiate into any fetal or adult cell type in the inner cell mass. So these right here, the pluripotent stem cells. We talked about multipotent stem cells when we talked about those hemocytoblasts. And so those are already a little more specialized and differentiated. They can only become blood cells. And so I just kind of wanted to tell you that there's different types of stem cells, and that has to do with the genes that are turned off or turned on, because once genes kind of get turned off and you kind of go down a path of differentiation, it's really difficult to coax them back up and to be less differentiated. They're really working on it. It's a hot topic of research, but it's really difficult to go backwards, and I just kind of wanted to point that out to you. Okay, so in the embryological period, we are going to go from basically a mass of cells to this very important pancake, which is called the gastrula, to something that looks human by the end of the embryological period. And so you can think about what's occurring during that time. We have a whole lot of mitosis going on. So obviously we went from that 32 cell stage for the morula, the 70 to 80 cell stage of an early blastocyst, and clearly we're going to get to something, at the very end of this process, of course, that's going to be a baby. So we have a whole lot of mitosis going on. The other thing, too, is that those cells are going to grow. They're going to increase in size. Cleavage, they don't increase in size, but we're going through mitosis now, so we're going to get cell growth. The other thing, too, we're not a ball. We're not a pancake. We have, appendages and things like that. And so the cells are going to migrate. They're actually going to move to resemble those external and internal form of the human body. And the two I just kind of showed you, they're also going to differentiate. So they're going to take on a specific structure and then, of course, a specific function. So we have a whole lot to happen throughout the course of pregnancy, but particularly in the embryological period. Okay, so we are going to kind of continue on what we were watching. It's so much easier. I realize I'm showing you a lot of YouTubes, but it's much easier to see in video format what's occurring.
Here we're going to go through quickly the early stages of development again. And then we're going to get to this stage when the germ layers are set aside. The three germ layers, the ectoderm, mesoderm, and endoderm. So here you'll see then that the inner cell mass gives rise to the entire adult animal. In this case, a human. This same process would occur in a mouse, as we'll see a bit later. If we now look at the embryo after the three germ layers have formed, this video will highlight what comes from the blue germ layer, the ectoderm. You'll see that it makes the nervous system, including the brain and the skin. The middle part, the mesoderm, green here, gives rise to the muscle, including the kidneys, the heart, and the endoderm gives rise to the whole gut tube. There you see the lung, the liver, the intestine. And to give one more detailed example of this, let's think about the development of the endoderm, and in this case, the formation of the pancreas. So there's the pancreatic bud, which comes out of the endodermal derivative. Now I'm going to talk to you a bit today about the pancreas as one example of how an organ and then the cell types within that organ get made.
Sorry, he's not. He's going to stop right there. But it was a nice kind of quick video for you. So this pancake formation with the 3 different colors, that's called gastrulation. And so the gastrula is a very important stage of development. Happens about day 16. And that gastrulation, that formation of those 3 primary germ layers, are what's going to be the first part of organogenesis. So we are going to get all of our organs being formed during this embryological period. So a whole lot of magic's happening. So let's look at this picture. And so this is showing you we've got sort of that implantation process. So this is going to be the endometrial side, and this is going to be the open or the lumen side of the uterus. And so it's showing you these 3 primary germ layers. And so ecto, so think outside. So think about the back side of you. So the first thing you come to is going to be your skin, you've got your hair on your head, and then you have your nails. So kind of envision anatomical position. Then of course you have that dorsal body cavity, which is going to be your nervous system, so your brain and your spinal cord. So all of that is going to come from the ectoderm. Think about that middle part of you, so the mesoderm. So we've got the circulatory system, we're going to get the epithelium, skeletal system, and muscular system. All right, so remember way back when in AP1 when you studied histology, remember connective tissue. So blood, bone, cartilage, all of those things were kind of lumped together, connective tissue proper, were all lumped together because they all came from the mesoderm. So think about mesoderm like the middle part of you and your muscular system. Endoderm, so inner. So think of all the things that are deep inside you. So that's, your digestive system, your liver, your pancreas, the inner part of your lungs. So just think of most of your abdominal organs. So from that endoderm layer. So when we have this sort of splitting into the 3 layers, gastrulation, we already begin the pathway of differentiation to get the different organs. And so so it's just kind of showing you those 3 layers. This happens about day 16, so really, really, really early. And then 2, once we get this differentiation of layers, that's when we're going to get those organs to be developed from there. So we're already kind of working our way to differentiation. So this is called a gastrula at this stage. So we've talked about a zygote, we've talked about just the period of cleavage, we get a morula, we've got implantation of blastocyst, and this is another really important stage in the embryonic period, gastrula. And so these cells are pluripotent. They can make human cells, but they're already specializing. Okay, so like I said, we are going to go from that mass to a pancake, if you want to think about it, to something that looks human by the end. So we're going to get all major internal organs taking place. We're going to also have to talk about the extra-embryonic membranes and the placenta. And like I said, you can see, ears, eyes, nose, you see fingers, you're going to see toes. You can kind of already see everything's in place. So the appearance of all major body structures, looks human by the end of the embryonic stage of development. Let's take a look at this picture. We kind of need to back up a little bit. So we're not going to talk about the embryo right now. We're going to talk about the 4 extra-embryonic membranes. And so we have the chorion, which is going to be the fetal part of the placenta. We have the amnion, which is going to create a nice sac for the embryo and fetus to develop in. And then we have the yolk sac and the allantois, which are going to become our umbilical cord. And so how this is going to happen, this picture is kind of backwards to the other one we're looking at, so I'm sorry about that. So we have that embryonic disc. We start off as that pancake we were talking about. And so this part, the epiblast, that's going to end up being the ectoderm. We're going to get the amnion. And then out here, that would be the endoderm, even though it's going to flip around. Like, so I don't really like this picture, but I didn't have another one. That's going to end up being the yolk sac. And from there, we're also going to get the allantois. So we're going to get the part of the umbilical cord from that. And so this is also showing you, because we're going to talk about placenta formation. I told you that trophoblast cells, they're going to become that chorion, that outer part. And there's two parts to it. We had the cytotrophoblast and the syncytiotrophoblast. And like I'm not going to ask you between those two, but you can see how they are just sort of digging into the endometrium. And then the mother's endometrium is going to send out so arteries going to the placenta. And so this is going to be a little group project when we go to make our placenta. But we're going to go look at these 4 extra-embryonic membranes. But I'm just telling you, they are also forming from those trophoblast cells and also from that bilaminar disc, so that developing embryo. There we go. All right, so we have the chorion, so this outermost membrane right here. We have the chorion, and then these are going to be what are called the chorionic villi. And so that's the fetal part of the placenta. So right here, chorionic villi. So outermost layer. And then this clear layer is called the amnion. And so that's shown here in the blue. What's going to happen is that the amnion and the chorion are going to fuse later in development. So we have this amniotic sac, and so this is going to be filled with amniotic fluid. So that's going to be about 100 mils of fluid. It's very similar to maternal blood plasma. And so this is just going to provide a nice cushioned and buoyant environment for the baby to develop. So it's just going to protect it. Two, as that embryo and fetus develop, definitely fetal development, what's also going to contribute to that is kind of gross, but you know, fetal urine as those immature kidneys are starting to develop. Two, as the fetus is inside that fluid-filled sac, it's also going to start to work on its breathing muscles, and so it'll actually sort of breathe in or suck in amniotic fluid and sort of exhale that. So that's the amnion. Let's see. Yeah, and so just kind of show you later in development. So we've got just those chorionic villi that are that fetal part of the placenta, and then we just had that outer amnion. And so you see that fused chorion and amnion about the 13th week, so in that fetal period. Okay, so now the yolk sac and the allantois, and so we're going to get this from that endoderm. And so you see this yolk sac, and then it's called the allantois, the other sac, it comes off of the yolk sac. I don't care. So the yolk sac is going to be on that ventral side, so the belly side of the embryo. So the important things about the yolk sac, that's going to be the first site of blood cell formation. We don't have bones yet. We don't have red bone marrow. So it's going to be first the yolk sac, then the liver, the spleen, and then finally we'll have functioning red bone marrow. It's also going to form part of the digestive tube. And then too, which is kind of weird if you think about it, this is also where the spermatogonia in boys and oogonia in females start to develop from that yolk sac. The allantois, yes, I'm calling it the other sac. This is going to be the functional part of the umbilical cord, and you kind of see where it is. And so it's going to form the 2 umbilical arteries and the 1 umbilical vein that are actually going to link this fetus to the placenta. So that's going to be the lifeline between mother and baby. And so this is kind of showing you that's the yolk sac. And then this is going to, here's the placenta, developing placenta over here. And then you see the umbilical cord right there. All right, so there are some types of genetic testing that can be done before the baby is born. So if a couple suspects that there's genetic abnormalities, they can do what's called chorionic villus sampling. And so remember those chorionic villi are those like finger-like projections. Since they are from those trophoblast cells, they have the genetic makeup of the baby. And so they can go in and they can just take some samples of cells from the chorionic villi, and then they could then test the DNA of the baby. Since this is done really early in a pregnancy, so the 10th week, there's a higher rate of miscarriage compared to the other one, which is amniocentesis. So this can be done 14th week of pregnancy on, so it's going to be the second trimester, but they can, using ultrasound, go in with a syringe and basically puncture that amniotic sac and draw out some of that fluid, because that fluid's going to have some of the fetal cells in it. And then they can just run those genetic tests, and then the couple, of course, could either just prepare for that or could choose to terminate the pregnancy. Okay, so just some highlights. I realize this is put out for expectant couples, but it's nice and short and kind of shows you everything that's going to occur during that embryonic period.
In the 4th week of your pregnancy, the ball of cells that is developing into your baby is the size of a poppy seed. It's at this stage that the cells separate into 3 distinct layers from which your baby's internal organs and skin will develop. The outermost layer forms the brain, nervous system, and skin. The middle layer forms the heart, blood vessels, bones, muscles, and reproductive system. The innermost layer will develop into the stomach, intestines, lungs, and other organs. Your baby's first blood cells are already being produced in the yolk sac. Expect big changes by the 5th week of your pregnancy. The ball of cells now resembles a tadpole, and it's as big as a peppercorn. The creation of the brain, spinal cord, heart, and blood vessels is already well underway. The neural tube, which will form the brain and spinal cord, starts to close from the center of the embryo outward. After it closes, the portion in the head thickens and divides into the forebrain, midbrain, and hindbrain. Rapid growth causes folding of the embryo. During this process, part of the yolk sac is incorporated into the lining of the digestive system. The primitive backbone is curled and looks like a tail, which will recede in a few weeks. Your baby's heart is transformed from a tube into an S-shaped loop that begins beating about 22 days after conception. At this point, you may begin experiencing early signs of pregnancy, including nausea, fatigue, and breast tenderness. By your 6th week of pregnancy, your little one has tripled in size and is protected by amniotic fluid. The baby has grown by leaps and bounds, but is still only about the size of a lentil. You've just been to the OB/GYN and she's confirmed the good news. You're definitely pregnant. You don't know if you're having a boy or a girl yet, but you can thank Dad for your child's sex, because one of 2 sex chromosomes, either an X or a Y, was delivered by the sperm determining your child's gender. The egg always contains an X, so if the sperm contained an X, the embryo develops into a girl. But if the sperm contained a Y, the embryo develops into a boy. A gene on the Y chromosome becomes active late in the 6th week, functioning for only a few days to tell the embryo to become a boy. Facial features have formed and his heart has a regular rhythm pumping fetal blood. Right now, because your baby's skin is only 1 cell thick, it's transparent. Little knobs protrude from the embryo's trunk. These buds of tissue will become arms and legs. At the beginning of your 7th week, your baby is now about the size of a coffee bean. The umbilical cord connecting your baby to the uterus is now visible. The placenta is covered with lots of small projections called villi. The villi from the placenta intermingle with your blood, where they steal some of your breakfast, lunch, and dinner. The nutrients and oxygen travel about 18 inches through the umbilical cord to your growing baby. It's too early to tell whether your child will have your nose, but mouth, nostril, and ear indentations are visible in the head. The arm buds that formed last week look like paddles. The brain is growing rapidly, including the right and left sides of the brain. The right side of the brain is associated with nonverbal and spatial tasks. The left side is associated with speaking and writing. You'll have to wait and see if your child becomes predominantly right or left-brained. By the 8th week, Your fetus is about the size of a blueberry. What were once just little tissue buds have become recognizable as hands and feet with webbed fingers and toes. Soon, this extra tissue between the digits will be reabsorbed to reveal individual fingers and toes.
Okay, so just got to see that. So yes, by the end, it's a little blueberry. How cute. But yeah, the disc rounds up. We're going to look at that formation of the neural tube is another really important part of development. The nervous system is going to be the first to start developing as far as organ systems. And then two, the nervous system continues to develop after the child is born. Like I said, by the 4th week, we're going to see limb buds. And then about day 20, the heart is noticeable and beating. So yeah, so that formation of the neural tube is called neurulation. And so it's just kind of walking through. We see our gastrula, so we have our 3 primary germ layers, and then it's going to start to fold. And what's going to happen is from that ectoderm, first we get sort of a neural groove, a neural ridge, and then it's going to be a tube. The thing is, the neural tube, and I don't have a great picture. I'm sorry, I was showing you the neural tube. The neural tube sort of zips up from the center towards the head, and then from the center towards the tail. And so sometimes this doesn't happen properly, it doesn't zip up properly, it doesn't close all the way. And so that's when you get spina bifida. And so this is just a better picture just showing you. So here's the neural tube. It's starting to zip up. And like I said, it goes towards the head and towards the tail. If it fails to zip up towards the brain end, and so that's called anencephaly. And so that's basically the baby would be born without a brain. If the bottom part doesn't close, so more the spinal cord, then it's called spina bifida. And there's several different types of it. But then you have, it's not encased inside of that vertebral foramen. And so they do know that there's a high correlation between folic acid deficiency and these neural tube disorders. And so that's why it's really important for women, if they're thinking about getting pregnant or during pregnancy, to get enough folic acid. I was just listening to the radio, which I hadn't realized. Usually this would be something that would have to be corrected after delivery. They can now correct spina bifida in utero, which is pretty amazing that they can fix that even before the baby's born. Okay, so now we're going to talk about the placenta and its many functions.
The placenta consists of maternal tissue, which takes the form of projections of the endometrium and fetal tissue, consisting of villi. The endometrial projections contain small pools of maternal blood, called sinuses. Maternal arterioles enter the sinuses and venules leave them. Each villus contains the venous and arterial vessels of the embryo. These are branches of the 2 arteries and 1 vein that run through the umbilical cord. The functions of the placenta include respiration, nutrition, and excretion. It transports water, oxygen, nutrients such as glucose and fatty acids, vitamins, ions, and certain antibodies from the mother to the fetus, and water, carbon dioxide, and urea from the fetus to the mother.
So let's talk about the placenta. So it's going to be sort of a group project. So it's going to be part fetal tissue and part maternal tissue. It's going to form at that site of implantation, and then it's going to continue to grow up until about 5 months when it's fully formed. And so this is delivered as the afterbirth, so this is a placenta. So just kind of think of it as a pancake. We'll look at another picture in a minute. But it is basically going to provide all the nutrition. So all the nutrients from the mother's blood are going to then go through the placenta into the fetal blood. Maternal blood and fetal blood do not mix. It's going to be through a membrane, which we're going to look at. All of the oxygen is going to come from the mother as well. And then the mother's blood will also be removing that waste CO2 from the developing fetus. It is also going to be an excretory organ, and so all of the waste created by the fetus are going to fuse into maternal blood, and then her kidneys will remove them. It is also going to be an endocrine organ, so the placenta makes hormones. So it's going to, at first, that developing placenta makes that human chorionic gonadotropin, But then the placenta is also going to be making estrogens and progesterone and placenta lactogen and a variant of human growth hormone. It's basically to regulate the metabolism of both the mom and the developing baby. And so just has really big endocrine function. So like the exchange of gases and nutrients and wastes between maternal and fetal bloodstream, no actual blood flow. So no blood flow between the two. So some problems with placenta formation. So ideally the placenta, because ideally we wanted implantation high up on the uterine wall. And so this is just showing a nice placement for the placenta. What can happen is what's called placenta previa, and that word just means placenta first. And what we don't want is the placenta to cross the opening of the cervix, because the placenta cannot be born or not leave the mother's body before the baby does, because that is the baby's lifeline. It's connected to the placenta by the umbilical cord, and that's how it's getting all of its nutrients and its oxygen. It's not uncommon to happen early in pregnancy, and as the uterus grows, it can kind of self-correct itself. But if late in pregnancy it is crossing this opening, then that mother would be put on bed rest. And then two, if it is across this opening late in pregnancy, that baby would have to be delivered by cesarean section. Think about it, as the mother moves around, the reason why they put her on bed rest is because there's a connection between the fetal part and then the endometrial side, so on the mother. And so if this is now across an open space, and as the woman moves around, those connections get severed, and so there can be bleeding. And two, the more connections that are severed between the placenta and that endometrium, that risks that baby being deprived of oxygen. And so that's why that mother's put on bed rest, to just restrict motion and that possible severing of the placenta from that uterine wall. And so yeah, placenta previa. So when the embryo first implants, it's going to get its nutrients basically from the endometrial cells, and oxygen and carbon dioxide can freely diffuse because the embryo is tiny. By the second month, the placenta is providing those nutrients and that oxygen. And so this, take a look right here. This is just kind of showing a cross section. So you've got out here, this is the maternal side, and it's just think about like sinusoidal capillaries. So we're going to get a rich blood supply from the mom, so to the decidua basalis, the maternal part of the placenta. And then we have these chorionic villi, and then we have these capillaries. So there's embryonic capillaries. And so diffusion is going to be across that placental membrane. And so you have the wall of the villus, you got some connective tissue, and then you have the embryonic capillaries. And like I said, these villi are just sitting in just rich vascular maternal blood. And so that everything good's going to come from the mom's blood, go into the fetus, and then all the waste are going to then diffuse from the fetal blood into those lacunae, those open spaces, and then the mother's circulatory system is going to remove those wastes. So when we look at the umbilical cord, so look over here, there are 2 umbilical arteries and 1 umbilical vein. And they're given their names, so arteries always go away from the heart in reference to the fetal heart. So we have 2 umbilical arteries going away from the fetal heart, picking up nice rich oxygen, and then 1 umbilical vein going back towards the fetal heart. And so I always kind of remembered it like this. You can draw like a little face. I'm sorry, when I'm in class, I write it down for you. So think of the 2 eyes like A's, and they're blue because they're carrying deoxygenated blood. And then the vein kind of looks like a mouth, so it's red. So 1 umbilical vein. So hopefully that'll help you remember that. So this is a cross section through an umbilical cord. You think how important the umbilical cord is, because it is that connection between fetal circulation and the placenta. And think about it, as this baby's developing, it's going to start to move. And so what can happen is what we don't want to happen is a knot in the umbilical cord. And we also don't want anything to crimp any of these vessels and prevent that connection between the baby and the placenta. And so here's another picture. So I'm showing those 2 umbilical arteries, 1 umbilical vein, and then what I want you to see, this is called Wharton's jelly, and it's just some magical connective tissue that actually prevents a knot from being formed in that umbilical cord. And there's really no way I can kind of I can say that to you, but I want to try to show it to you. So here's another quick. This is a placenta, so it's delivered as afterbirth after the baby, but she's going to show you just the magic of Wharton's jelly.
This is a placenta party trick. This is a knot in the cord. And you can see, you can slide it up and down the cord. The cord has Wharton's jelly in it on the inside, and it keeps it from constricting when rolly little babies create knots in their cord. So this is not a true knot, not a true knot. A true knot would be where it constricts a little bit tighter and doesn't slide up and down on the cord. Cool, huh?
Okay, so like I said, that's the function of that Wharton's jelly. It protects the umbilical arteries and vein and then prevents those true knots. And sometimes true knots do form, and then of course it can kill the baby. All right, so if you think about it, since the baby is getting all its oxygen from the placenta, so from the mother, there is really no reason to send that blood, that oxygen-rich blood coming from the placenta to the fetal lungs, because the fetal lungs aren't developed yet, and the fetus is developing inside amniotic fluid. So there's two bypasses to the pulmonary circuit. And so that wall in between the atria, there's something called the foramen ovale. And so it just means oval-shaped hole. And this is something that will become the fossa ovalis, if you remember that structure. I know it was a long time ago we learned that, before 1 year. There is another bypass called the ductus arteriosus, and it's hard to see, it's right there, but it's between the pulmonary trunk and the aorta. And so it's going to become the ligamentum arteriosum, if you remember hearing that, about 15 minutes after birth. And so once again, you've got to think you've got that umbilical vein that's going to be oxygen-rich blood, and there's no reason to send all of that to the lungs. The lungs are going to get some blood from that left side of the heart, but there's just two bypasses, so the blood goes from the right side to the left side and then to the body.
The placenta, located in the mother's uterus or womb, allows the passage of oxygen and nutrients from the mother's blood through a membrane to the baby's blood. From here, it flows through the umbilical vein into the baby. With the lungs collapsed, there is no need for the right side of the heart to send blood to the lungs. The blood is already oxygen-rich thanks to the placenta. Instead, there are two shortcuts that allow the blood to bypass the lungs. The ductus arteriosus is a structure that detours blood normally routed to the lungs and lets it flow straight into the aorta and on to the rest of the body. The foramen ovale is actually a hole in the middle wall or septum of the heart, which allows blood in the right side to flow through the wall into the left side and out to the rest of the body. In both cases, the lungs are bypassed. Finally, the ductus venosus, which is a continuation of the umbilical vein, serves as a shunt or bypass, which allows most of the blood returning from the placenta to be diverted from the immature liver of the fetus and emptied directly into the inferior vena cava.
Okay, so maybe remember we looked at that fossa ovalis at the beginning of the semester when we studied the heart. And so during fetal development, this is open, so the foramen ovale, and that's just so that oxygen-rich blood coming from the placenta can just bypass over to the left side of the heart and then be sent to fetal circulation. So that is one of the bypasses. Sometimes, though, the foramen ovale doesn't close, and so that's called a patent foramen ovale, and it is an atrial septal defect. They estimate it's about as high as 25% in the population, so it is something that's very common. It just depends how open it remains, if it's ever detected, or two, it can also be detected sometimes if someone is like a marathon runner or really putting a high demand on their heart. They may have problems, and then it's detected, and so then it can also be corrected. But that's what they mean when they say a patent foramen ovale. It is that atrial septal wall defect. And then the ductus arteriosus, that is, here's a better look. So between the pulmonary trunk and the aorta, quickly after birth, that is going to close and become the ligamentum arteriosum. And so that's just another bypass of the pulmonary circulation. And this is just kind of showing the bypass to the fetal liver. And if you think about it, remember all that drainage from the intestine goes to the liver to be filtered and detoxified. Mom's taking care of all of that. And so we have that bypass, the ductus venosus. It's going to become the round ligament of the liver and ligamentum teres hepatis. And then two, after birth, those umbilical arteries just also become ligaments. And so umbilical ligaments, because they're no longer needed. And so the belly button, that is basically a scar from where the umbilical cord was. So you think about, remember when we studied respiratory, that oxygen and gases, they just freely diffuse. And so think about this, once again, it's chemically, we have to get oxygen unloading now from the maternal red blood cells to the fetal red blood cells. And how are we going to get this done? It's kind of a nice trick. Fetal hemoglobin has a higher affinity, about 20 to 30% higher, higher affinity for oxygen than adult hemoglobin. Two, fetal blood also has more of these hemoglobin molecules. So that's sort of how the fetus can steal that oxygen basically from the mother. Just other things about the placenta. We know that antibodies, so those IgG antibodies, can cross that placental membrane. Two, most drugs can cross the placental membrane as well. And so we'll talk about things that are contraindicated against during pregnancy and for breastfeeding. Also, there are some diseases that can also cross the placenta, so syphilis, toxoplasmosis, listeriosis, German measles, HIV. And two other diseases can be passed to the fetus through the birthing process, so things like herpes, other FTDs, gonorrhea, chlamydia. Those are other things. So it's great that there is no mixing. I just kind of want to make sure you got that part. There's no mixing between fetal blood and maternal blood until that birthing process, and that's when we learned about, remember, erythroblastosis fetalis or hemolytic disease of the newborn. That's when that's when there's that mixing of blood and introduction of the fetal blood to the mom. So hopefully you kind of remember that. All right, so I'm going to stop part 2 here and come back for part 3.